Assalamualaikum and hi everyone. Our title for our presentation is Gravimetric Analysis, focus on pharmaceutical analysis on how to determine the concentration of chloride ion in seawater sample. So this is our team members, consists of me, Wan Nur Christina binti Wan Jamri, Nur Atirah binti Asma Peter, Nur Ain Nabihah binti Muhammad Khairul Nizam, Nur Faisha Erika binti Nur Azian, and lastly, Amirah Adani binti Ahmad Zaidi. So this is the content for our video. Conclude introduction, principle of gravimetric analysis, types of gravimetric analysis, methodology, example of result, and lastly conclusion. So our main title in this video is about gravimetric analysis. So what is gravimetric analysis? Gravimetric analysis is a compilation of qualitative analysis laboratory methods based on mass measurement of an analyte. And what is pharmaceutical analysis? Pharmaceutical analysis is an analytical technique used to determine the quality and quantity of the pharmaceutical products. How to determine the chloride ion in seawater sample? We can determine the chloride ion in seawater sample by titration using silver nitrate as titrant and seawater as analyte. The analyte need to add a few drops of indicator before titrate with the titrant. The chloride ion is present in the sample if the drops land on the sample and produce white precipitate.
So next is the principle of gravimetric analysis. The principle of gravimetric analysis is based on the estimation of the mass percent of an ion in an impure compound of known quantity by determining the mass of the same ion in a pure compound. Thus, based on the title of determining the concentration of chloride ion in a seawater sample, we can measure the amount of silver nitrate needed to react with all the chloride ion in the seawater. Thus, we can calculate the chloride ion concentration.
Okay, my name is Nafarsha Erika Bentino Azen. I will explain the steps that have in methodology. First step is sample preparation. The sample is first collected and prepared for analysis. The sample should be sure of the overall sample population and should be homogeneous. This may involve grading or crushing the sample to ensure homogeneity. Subsequently, precipitation of precipitate. The analyte is converted into a solid precipitate by adding a reagent that forms a sparingly soluble compound with the analyte. This reaction should be selective and quantitative, ensuring that only the analyte of interest form a precipitate. Next, filtration. Filtration is the precipitate that separate from the solution by using a suitable filter paper or other filtration apparatus. Next step is washing. Washing is the precipitate washed with a suitable solvent to remove any soluble contaminants. This step is crucial to ensure that the purity of the precipitate. Next step, drying. Drying is after washing, the precipitate is dried to remove residual moisture by using oven. Weighing is the dry precipitate is weighed on a balance to determine its mass. What I was trying to say, the weight of precipitate is then determined by the difference between the final weight of filter paper with precipitate and initial weight of filter paper. Okay, last but not least, is calculation. Calculation is the last step of methodology. Calculation is the amount of analyte present in the original sample is calculated using the weight of the precipitate and the stoichiometry of the reaction. Okay, I will pass to the next slide.
Next, example of result. The title for the experiment is Determination of the concentration of chloride ions in a sample by gravimetric analysis. Objective for the experiment is to determine the concentration of chlorides in seawater sample using gravimetric analysis. This is introduction for this experiment. So this is apparatus, chemicals, and materials that we use to conduct this experiment. Next, for procedure, we have 4 stages in this experiment, which is sample preparation stage, precipitation stage, filtration and washing stage, and lastly, drying and weighing stage. So this is 4 stages that we use in this procedure to conduct the experiment. For part calculation, based on the result, we can calculate the mass and construct the gravimetric factor. So once the gravimetric factor of the analyte has been obtained, the single mass of the analyte can be extracted from the mass of the precipitate. Now, the concentration of ion in the sample seawater can be calculated by using the following formula. Discussion for this experiment. The experiment conducted demonstrates the efficiency of gravimetric analysis in finding the concentration of chloride ions from a sample of seawater. In order to calculate the concentration of the ion, 2 different salts were used in an ionic replacement reaction to produce 1 solute salt, sodium nitrate, and 1 insoluble salt, silver chloride.
So, in conclusion, the concentration of chloride ion in seawater sample can be successfully determined with this method, which is gravimetric analysis. By this method, the content of chloride ion can be obtained by all procedures starting from preparation, titration, and form the precipitate of AgCl until the calculation part. So, based on the example of results, the mass of chloride ion in 100 ml sample is 1.94 gram. So through the preciseness of the result, it is also show how gravimetric analysis is useful and accurate for monitoring the concentration and the mass of ion present in sample. Moreover, the principle of solubility also were observed, and the stoichiometry was used in the experiment for the calculations. By this function, we can apply to monitor the concentration of chloride ion in seawater to protect marine life and the environment, since the high level of chloride and salt concentration in water affect the distribution of aquatic plant and animal species according to the amount of salinity they can tolerate. Therefore, determination of the salt content of seawater is an important area of research, since the ocean currents and global climate are affected by salt content. So, based on the article also prove an excess of salt can have an impact on saltwater habitat as well. There is a limit to the amount of salt that local plants and animals can withstand. They risk dying or suffering are the negative effect if there is an abundance of chloride or salt. Then seagrass, microalgae, and phytoplankton, as well as their ability to photosynthesize, can be negatively affected by such salinity shift, which might have an impact on the variety and metabolic potential of coastal microorganisms, as well as the global biogeochemical cycle. That's all from us.